one of my other lecturers already did Unit 5, but I hope that they posted the recording for that. What units have I done? I've done Unit 7 and I've done Unit 6. And I'm in the process of doing Unit 6. The recording, it's on the lecture slides. Check the announcements posted for 5 Hive. Oh, sorry. No. Okay. Yeah, I'll cover this. I'll cover all these questions. Thank you. Is Ligand and DNA replication same as the one? Okay, so I think that you're confusing Ligand with Ligase. Oh yes, I will post the slideshows later. But yeah, back to Kitsen's question. I think you're confusing Ligase with the Ligand. Ligase with an S, I'll spell it like this. Ligase, I actually see the confusion. Actually, I see, yeah, this is pretty confusing. Ligase is for DNA, while Ligand is for the, yeah, I'll cover this all later. Oh shoot, we got Lance in here. All right. I'm locking in Lance. I'm always locked in.
No, wait, sorry. Just to interrupt, it's actually ligase, not ligase. Actually.
Okay. All right. Thanks, Lance. All right. All right. So let's get back on topic. Oh yeah. Lance is actually my master. Yeah, he's my master. Yeah. All right. No, all right. No pause. We're gonna play this. We're gonna play this video. Ligase. All right. I'll keep that in mind. Tell me if you guys. I'm certain. I'm playing this, but let me know if you guys can't hear the audio.
Sequence of 4 twisted around. DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process.
Shoot, is the video like playing at like 2 FPS for you guys? Is it watchable? Oh, okay. Okay, 20 FPS. Okay, all right. Don't worry, guys. I'm playing this video just to give an overview, and then I'm going to cover everything that's in this video more thoroughly in depth after. So it's not. I hope to. Yeah. So there's only 2 minutes left, but I'll cover everything. Yeah.
This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, so hopefully that video wasn't too choppy for you guys, but if not, then I'll be covering everything again. And again, I see some questions. Let me open
DNA is a molecule made up.
All right, so, what's Ligase's job? All right, I'll cover that right here, guys. All right. So that video just had, guys, I recommend this YouTube channel. They're called Your Genome. They have amazing animations. They helped me so much, like years ago when I was learning, and they're still popular today. But all right, let's get started. All right, why is this not going? What? Oh, okay. All right. So, keynote about DNA replication, and this is the key. This is the key. All right. So, oh yeah, I'll totally do that. Yeah. Again, I'm just posting the CD in the chat. Don't get too confused by it. I'll cover everything in it, but I just like putting it there for just overview purposes. All right, so back to the keynote about DNA replication. So one very important point you need to know about DNA replication is that it's semi-conservative. So what does this mean? So this means that each new DNA molecule consists of one original parent strand and one newly synthesized strand. In other words, you can conserve one old strand and make one new strand. So let's look at this diagram. So look at blue. Blue is the parent strand, and red is the. All right, I see a bunch of questions in the chat. I'll cover it after I finish the slide. All right, so blue. Let's say blue is the parent strand, and red is the child strand. As you can see, it's semi-conservative because it starts with one DNA strand, but it ends in two, and each one has a parent strand, and each one has a red strand. And the red strand indicates the two daughter strands. And so that's why we say that DNA replication is semi-conservative, because each daughter strand is comprised of one parental strand and one new strand. All right, any questions about that? All right, let me catch up with Discord. All right, what's DNA again? So DNA is the main heritable molecule inherited by all life. It transfers genetic material from one organism to another. And DNA is comprised of a deoxyribose sugar and alternating phosphate and deoxyribose sugar backbones. And yeah, that's just pretty much DNA. All right. What was the red strand? So the red strand is the daughter strand. The red strand is the newly synthesized strand based off of the parent strand. All right. So I'm just going to answer the questions in order. Do we need to know the other types of conservation? No. Which of the daughter strands are the exact same, so how is it not fully conservative? Well, the reason the name came from semi-conservative is because it's not. If it was fully conservative, it would just be one. It would just be a duplicate of itself. So, sorry. Let me think about how I'm going to explain this. It's semi-conservative because one daughter strand is made off of the original parent strand. Actually, let me get back to you on that one. Do we need to know about DNA replication in prokaryotes, or is it the same? Back to that later, but DNA replication in prokaryotes is pretty much the same, except it's done simultaneously as translation, which I'll cover later. What was the red strand, the new strand? Is ligase only for the lagging strand? Yes, and I'll cover that in a sec. So, back to your question, Moose. Basically, the daughter strands are the exact same as the original ones, so how is it not fully conservative? So I would say that the term semi-conservative in this case came from the fact that only one strand was maintained. So yeah, some is saved and some isn't. Yeah. All right. Yeah, sorry. I'm still catching up with all the Discord questions. So can you explain what semi-conservative is again? Okay, so basically, Semi-conservative means that one single parent strand, which is the blue part right here, unravels itself into two separate strands, and using DNA base by base pairing, because remember guys, this is also an important thing, adenine only pairs with thymine, cytosine always pairs with guanine, of course, thymine uracil. And so, a second strand of DNA can always be made off of the parent strand of DNA using base by base pairing. So that's how these red strands are made, and that's how these are identical. So the base pairings are, oh yeah, let me know if that helps, Ruhi. So the base pairings are fully conservative, but it's semi-conservative with how the fact the other strand, but it's semi-conservative with respect to how the other half of the strand is being replaced by a new, yes, exactly, yeah. Yeah, what you have there is, yeah, that's right. Let me think. Sadly not in this session. Sorry for that, but I think that should be next session. Yeah. All right. Any questions about this before I move on to the next slide? I know it's pretty confusing, but okay. Just to review, DNA replication is semi-conservative. It starts with one parent strand, which separates into two separate strands, and using DNA base by base pairing, two daughter strands are made off of that, and so each new strand has a parent strand and a new daughter strand. All right, so the original fully splits, and the two formed DNA strands pair with their respective, yes, exactly, with their respective nitrogen bases. Let's say that, if you guys can see my red, this blue one right here is adenine, then this red one right here has to be thymine. All right, wait, so how does mitosis produce two genetically identical because the DNA has a new strand? Oh, it's, yeah, so, because, so how does mitosis produce two genetically identical because the new DNA has a new strand? So, this works because, I see the confusion, yeah, they're still identical. Yeah, I totally see where you're coming from. So, it's new, but, yeah, exactly, it's identical. So, although a new DNA molecule is made, they're still identical because of base by base pairing. So, let's say, okay, to take a look at my laser pointer right here, let's make this top one adenine, and let's make this top one thymine. Now, as this thymine molecule, let's say call this thymine, and let's call this adenine. Let's move this thymine molecule right here, right? And let's move this adenine molecule right here. Because of base by base pairing, oh shoot, am I pronouncing it adenine incorrectly? Shoot. Alright, but anyways, thymine will go here and adenine will go there. So these two are identical because of base by base pairing. Yeah, so sorry, does that make sense? Adenine? Shoot. Alright, sorry guys. I'm mostly a textbook type of guy. Adenine. Adenine. Oh my god. Adenine. Alright, guys, we don't talk about this. Adenine. Alright, anyways, back to base pairing. Adenine, thymine, so that's why they're the exact same. Sorry. Oh, I just got pinged. Oh, alright. So does that make sense? Let's see whose question I originally asked. Sorry, I can't find the original question, but does that make sense? Okay, perfect. Adenine. Alright, adenine. I got it now, guys. Alright, if there's no more questions, I'm gonna skedaddle on to the next slide. Alright, let me check Discord one more time. Alright, let's go. Alright, oh wait, I see there's a question. Oh, yeah. Well, here's the thing. I mainly learned AP biology through, I didn't have a formal teacher. I was mainly reading it from a textbook. But don't worry, guys. I'm still qualified, guys. But, alright, so back to this. Alright, so DNA polymerase and the 5' to 3' synthesis. So, DNA polymerase is the enzyme that builds new DNA strands. And that's the core. So, and it can only add nucleotides to the 3' end of a growing DNA strand, which means replication always occurs 5' to 3'. And I'll talk about this next slide, what exactly 5' to 3' is. And the most important thing is that here's, someone was asking about primers earlier. Again, DNA polymerase starts replication based on primers. And the primers are just RNA. You don't need to know much about RNA primers. I looked at the CED. all you need to know is that they signal the start of DNA replication. Like that's all you need to know about them, because they are natural biology. Yeah. All right. So. Okay. All right. Thank you. All right. Don't worry. This is a part 2. Guys, if this goes beyond 8:30, I'll be so sorry. But all right, let's go. All right. So, why 5 prime to 3 prime? So this is probably the most confusing part of this subtopic that most people get tripped off on when I've lectured on this in the past. So here's how it works. Each carbon in deoxyribose is labeled 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So, what does that mean? So, turn laser pointer. Okay, so as you can see, each carbon, this is 1, this is 2, this is 3, this is 4, and that's 5, right? So, and each carbon is labeled 1 prime or 5 prime, right? And the 5 prime end has this phosphate group right here, while the 3 prime end has the hydroxyl group. And so that's why nucleic acids can only be added onto the 3 prime OH group because of its hydroxylness. Does that make sense? All right. So sorry. Do we have to fully understand why 5 prime to 3 prime, or can we just remember? Okay. Here's the thing. You don't need to. I looked at the CED. You don't need to understand why. You just need to know it's. Can you back one more time? Yeah, of course. So you don't need to know why 5 prime to 3 prime. You can just remember that it's 5 prime to 3 prime. That's the synthesis. But yeah, I whenever I'm doing stuff, it always helps me to understand why, and that helps me know why, if that makes sense. But yeah, you don't need to know why. You just need to memorize why. But yeah, all right. Any questions about that? All right, I move on. All right. So here's the second most confusing part. Actually, wait. Any questions about this? All right. I don't want to hold up you guys too much, but all right. So the leading strand. So, all right. Sorry. So 5 to 3 means new nucleotides are added on the 3 side, right? Yes. 5 prime to 3 prime means new nucleotides are always added onto the 3 prime end of DNA. All right, let's move back. All right, I see there's some questions. So wait, I zoned out. Why is the number of carbons? you're so good. You're all good. The number of carbons are important because each carbon in the deoxyribose is labeled 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And DNA is always synthesized 5 prime to 3 prime because new nucleotides can only be added onto the 3 prime end right here. All right. Being able to understand the lag strand. All right. Yeah. It's all good. Yeah, all good. All right. Time to get on to the actual big part. Do you need to know P group and hydroxyl group? No, you don't. Well, I mean, I personally think, yeah, you won't be tested on it. Okay. You need to know that you need to know that it's made of a phosphate group and hydroxyl group, but you don't need to know why it's 5 prime to 3 prime. If that makes sense. Yeah. But you do need to know, it's essential knowledge that you know that DNA is made up of alternating phosphate and deoxyribose backbone. But yeah, that's what you need to know. All right, so back to this. So the leading strand is the easier side to replicate. It's synthesized continuously in the same direction that the replication fork is moving. Now, if you guys remember from the video, here's the thing. Here's the dilemma that the evolution masters had, right? DNA can only be synthesized 5 prime to 3 prime, right? So how is it possible that we can synthesize the lagging strand going backwards? So I'll cover that. I'll cover the lagging strand in a moment. But essentially, the leading strand is the easiest strand to synthesize. Helicase unwinds the DNA, and the leading strand synthesizes DNA from a 5 prime to 3 prime, and it can do this continuously because you can just follow the replication fork. And I'll cover the lagging strand in a second. All right, what's the difference between the coding strand and the leading strand? I'll cover that. Actually, I'll cover the coding strand and I'll come back to this question. Yeah, all right. So here's where it gets hard. So the lagging strand. So the lagging strand is a little bit trickier, right? It's not a little bit trickier, it is a lot trickier. It's a hard concept to wrap your head around. So the thing about the lagging strand is that it's synthesized discontinuously. So what does this mean? What does discontinuously mean? So like I said, oh shoot, it's built in small fragments called Okazaki fragments. So what are Okazaki fragments? Well, as the DNA gets unwound by helicase, there's two strands that get made, right? This strand, which is called the leading strand, and this strand, which is called the lagging strand. And so as helicase unwinds the DNA here, the leading strand is able to synthesize easily because this end is 5 prime, and this end is 3 prime. So it's able to do this very easily. But for the lagging strand, this end is 3 prime. Oh wait, it's labeled 5 prime right here, but that's for this strand. But this specific, where the end of this arrow right here, that's 3 prime, and where this is right here, that's 5 prime. So in order to synthesize from a 5 prime to 3 prime direction, it does it in reverse. So take a look at this. This is DNA polymerase. This is also DNA polymerase. I'm actually getting my pen out for this. So this is DNA polymerase, and this is also DNA polymerase. So what happens is that this is 3 prime right here. Sorry, no, 5 prime. This is 5 prime right here, and this is 3 prime right here. And the Okazaki fragments get synthesized from a 5 prime to 3 prime end. And it does this in reverse. And here's the thing. It makes these fragments called Okazaki fragments because it has to do it in segments. And these fragments are later joined together by an enzyme called ligase. Ligase. I got it right this time, guys. But it acts as the molecular glue joining these fragments together. So here's what I'm talking about. It has to do this in segments, right? So when it reaches the end right here, like let's say a polymerase starts right there and goes right there. Ends right there. The ligase is the glue that joins these fragments together. All right, so that's the lagging strand. Let me open Discord. I'm pretty sure I have like 1,000 questions. Let me look. Wait, is there any audio? Oh my gosh, was I muted that entire time? Okay, no, yes. Okay, perfect. Okay, sorry guys. I'm only checking Discord. Like, one monitor is a little pain. All right, let's check. What's the difference between the template strand and the? All right, I'll cover the template strand and the non-template strand in a sec. Next slide. So does it basically take sections of the lagging strand to prime and add bases and then get joined back to by ligase? So you're saying that does it basically take sections of the lagging and yes. So the primers get made and then bases get joined and they get joined by ligase. So your crest, what you have right there is right. The primase is jumping backwards on the lagging strand. Well, by backwards, I'm not too sure what you mean by that, but primers are being added to signal replication. Like towards 3 prime. Yes. Wait, sorry. Am I understanding your question correctly? 3 prime at the bottom. Like, you're talking about this? Is it jumping backwards? Okay. Well, actually, in theory, yeah, no, it's going to 5. It's going to 5. In theory, this primers will prime this part first, then this part next, and this part next, this part next, this part next. Yeah. All right. So wait, why is the number like switched again? Like I don't know how to describe it, but the numbers are switched at the top left. Okay. Yeah, that's totally fine. Yeah. This was very confusing to me when I learned it last year. So here's why. So it starts with the 3 prime end, and let's just follow this. Look at my diagram. We can just follow this. This is 3 prime end, and we can just keep following this. As you can see, it gets 5 prime right here, right? And here's why the numbers get switched. It starts because DNA is anti-parallel, meaning the strands go in opposite directions. This has to be 3 prime, and this has to be 5 prime, and that's just the I like to say definition of DNA, because they're anti-parallel. So the numbers switch because this is 5 prime and 3 prime, and then we can keep tracing this. 3 prime, 3 prime, 3 prime, 3 prime, 3 prime, and now this ends in 5 prime. And the reason the number gets switched is because now this has to be 3 prime. So is this what you mean by what the numbers get switched? Sorry, let me look. Does that make sense? Hey. Oh yeah. All right. So it's OG strand. Wait, sorry. Is the orange strand DNA, RNA? Okay, the orange strand is DNA. But I see where the confusion comes from because I did say RNA is single-stranded, but the orange strand is DNA. The orange strand is DNA. The orange strand is indeed DNA. All right. since the lagging strand is so hard to understand, it goes into an uphill direction of 3' to 5'. Yeah. Oh yeah. Why is there RNA at Okazaki fragments? Okay. I think I can't see that thing that you replied to. It says no message, but the RNA at Okazaki fragments would refer to the primers that signal the start of replication. All right. Oh my gosh, I didn't realize RNA was in here. RNA. Okay, so RNA is not in this diagram, but you should always know that RNA primers always signal the start of DNA replication. It's not on here, but you should always know that RNA primers always come before DNA replication. Sorry. No, well, okay. No, RNA polymerase is making... DNA polymerase replicates DNA. Right here, right now, is just... This is DNA replication. This is DNA replication. So it's only DNA, no RNA. Yes. Okay. In a sense, yes. This is all DNA, but okay. Sorry, I don't want to confuse you more, but back to this side. Shoot, where was it? Okay. But RNA primers always come before DNA is replicated. They signal the start of replication. If that makes sense. Okay. Yeah. Can you get a few questions wrong? So yes, you can get. The curve is, guys, AP is, okay, sorry, no, I can't get on a tangent like I did last week. But yes, you can still get a few questions wrong and still get a 5. You can get a lot of questions wrong. You can get like an 80% and still get a 5. Did we go over the different DNA polymerases yet? No. Okazaki fragments are from 3' to 5'. DNA is always from 5' to 3', and Okazaki fragments are simply segments of DNA. So the Okazaki fragments are from 5' to 3'. Again, look at this right here. This is 5', this is 3'. Because Okazaki fragments exist because DNA has to be synthesized 5' to 3'. So DNA polymerase has to do in reverse order. RNA primers, why not DNA primers, primase? Okay, so the reason not DNA primers is I'm honestly not too sure myself, but RNA primers are that's just how the cookie crumbles. Yeah. So the RNA primers start DNA replication, bind the old DNA, and they get replaced by DNA polymerase with a new DNA strand. What you have there is right. Yes, what you have there is right. Guys, it's all good. Guys, this is why I split Unit 6 up into 2 sections, because I knew how confusing it was. So we're halfway there though. So, guys, yeah. Do you wear wigs? I do not wear a wig. Yeah, I don't wear wigs. Do I like cheese? No. I love cheese. All right. Wait, sorry. Sorry. You still have said this, but why are there fragments? Because isn't it going 5? Okay. So that's a good question. So Okazaki fragments, again, DNA has to be synthesized 5' to 3', right? This is the 5' end. This is the 3. Sorry, I'm just gonna start with 5' end. This is the 3' end, and this is on this strand right here. This is on this strand right here. But here's the thing: DNA. Let's say this is DNA polymerase. It can't go this direction, right? Because in that case, that would be 5' to 3'. So DNA polymerase has to go this way, and has to do it in fragments, because it's going in reverse. If that makes sense. Does that make sense, Ari? All right. I'll answer some questions. Roughly. Yeah, sorry. Yeah, it is. I don't. Okay, perfect. All right, guys. Next slide. You guys, I'm mostly fine with answering all these questions. Although I know some people have to be there. Again, slideshow will be posted. Recording will be posted. I have to do something. Yeah, guys, just pass the ball at that point. All right. Oh, this is the slide I've always. All right. So back to topoisomerase, guys. Oh my gosh, no. Don't do this to me right now.
Topoisomerase.
Okay, what does topoisomerase do? All right, topoisomerase is another important enzyme. So, as the helicase unwinds the DNA, it causes the DNA ahead of the replication fork to become supercoiled and stressed. So, what is supercoiling, right? So, here's my analogy. It's a bit. The formal definition of supercoiling is a tension state ahead of the replication fork. But here's how. That's just a lot of mumbo jumbo. So let's say that you have a USB-C cable and it gets tangled. in order to easily unraveling that is essentially undoing the supercoiling. So, topoisomerase solves this problem by okay, formally, you don't need to know this. It cuts the DNA, relaxing the tension, and then rejoins it. But you don't need to know that. All you need to know is that this enzyme, that topoisomerase, unwinds supercoiling and relaxes DNA ahead of the replication fork so that DNA replication can proceed smoothly. As you can see right here, this is the replication fork, this is topoisomerase, and it unwinds it nicely so that helicase can unwind it. And yeah, so let me open Discord. So what do I mean by supercoiling? Supercoiling is the tension state ahead of the replication fork. It's essentially this twisted state that's so hard for helicase to unravel. A helicase, it forms the replication fork by opening out the DNA double helix. Alright. Yeah, I understand. Yeah. Alright. Does helicase not affect these or what? Sorry. Oh, this part right here? Well, helicase is ahead of the replication fork, so it's not even touching this. Oh, you're saying why those... Because they're after the helicase. As you can see, like, the helicase is going this way. Wait, sorry, let me just restart. Helicase is going this way, right? Oh, yeah, all good. Yeah, and so it's not touching this because it already did. If that makes sense. It doesn't touch it twice, yeah. So topoisomerase is ahead of helicase, which is ahead of the two DNA polymerases. Yes. Yeah, that's a good way to put it. Can you real quick go through the order of these enzymes? I'm gonna go over the order of these enzymes at the end. I have a review slide at the end, and I'll go over those again. So helicase and topoisomerase both do DNA unwinding, but in different spots. Okay, helicase does DNA unwinding here. It does different types of DNA unwinding. Helicase splits it. Okay, this is helicase. This is topoisomerase. Helicase actually splits it here, and this is what I mean by unwinding, while topoisomerase just simply makes it look smooth like that. Yeah, of course. Alright, any more questions about this? Do we need to understand upstream? Okay. No. Well, I checked the CD just now. Wait, what do you mean splits it? Okay, this is helicase. This triangle, this is helicase. Yeah. Heli, and it splits it by... This is what I mean by splits it. It splits it in half like this. What the hell, Elon? Alright, sorry. Breaks hydrogen bonds. Yes, exactly. Yeah. Helicase is to Moses such that DNA is... Yes, possibly by C. Is helicase breaking the hydrogen bonds? Yes. Yeah, it is breaking these hydrogen bonds right here. It is breaking the hydrogen bonds right there. Alright. Yeah, everyone, alien, that's right. Yeah. What the hell, Barry? What type of conceptual questions will they give about this? I've taken... Oh, yeah. When reviewing for this lecture, I only took like 2 practice exams, but in the questions I saw, it will be a direct MCQ and just testing your knowledge on this. Just your raw rote memorization. It's gonna be like, what enzyme unwinds the DNA? Sorry, what enzyme splits the DNA and forms the replication fork? Answer will be helicase. What enzyme relaxes supercoiling? You're gonna know that's topoisomerase. Yeah. So it's just, yeah. I mean, I've only done, guys. Again, I had to review for this, so I've only done like two practice pooling FRQs. But in the FRQs I saw, it wasn't really tested on this. FRQs are mainly conceptual, like analyzing data, trying to find how the crosses go. But I've only seen mainly MCQs. All right, so I saw a question like the last slide, and it was asking which way the 2 arrows go. This side. Wait, sorry. Do you still have a question about that? All right. Because all right. If you still have a question, you can ask me again. All right. Next slide. No, I was asking what type of questions. Oh, for this right here. What the? Oh, right here. Oh, okay. So they're gonna ask you. They might ask you. Okay. So this I've actually have seen on an FRQ. I do vividly remember. That it's gonna ask you to explain Okazaki fragments. And it's going to ask you to explain why do Okazaki fragments exist, and why are they formed in 3' to 5' direction. And that's something they might ask. Alright, I'm going to skedaddle. Yeah, no problem. Alright, guys. I'm currently in the darkness. I'm going to turn the light. Oh yeah, of course. Yeah. Alright. Okay, so this is the review. Again, this is the review. These are the 4 key enzymes you need to know. These are the 4 that will be most tested. Helicase, it unwinds the DNA. Topoisomerase, relieves supercoiling. DNA polymerase, synthesizes new DNA. And ligase, it joins the Okazaki fragments. These are the key enzymes that you need to know. These are the key enzymes you need to know. Alright, so, and actually, because there were a lot of questions about primase earlier, I do want to add a little box about primase. It signals the start of of DNA replication. Alright, so here's the summary. Here's the full summary of everything that we just learned. DNA replication is semi-conservative. That means that in 2 daughter cells, 1 strand is from the parent, 1 strand is from the daughter. DNA is synthesized 5' to 3'. We don't need to know why that is. You just need to know 5' to 3'. The enzymes are helicase, topoisomerase, polymerase, and ligase. And these are the enzymes you will need to know. Right, sorry. This is not really an enzyme. But I just add that there just now. And a leading versus lagging strand. So, again, this is the concepts. And any last minute questions before I give the AP MCQ? Alright, let me check Discord. Alright, do we not need to know RNA primase? You do need to know it, but it's not going to be heavily focused because it wasn't actually mentioned in the CED. What about the order? Yes, back to the order. Here's the order. It goes helicase, topoisomerase, and sorry, let me revisit your question. Sorry about that. I said I would revisit it, but I didn't. Yeah, so it goes helicase, topoisomerase, and the 2 RNA polymerases. Oh, no, sorry. First of all, is the 2 RNA polymerases. Sorry. This is 1 right here. This is 2. This is the helicase. 3. This is topoisomerase. Does that make sense? Oh. Okay, wait. Yeah. Okay, so from left to right, according to this diagram right here, it goes DNA polymerase, then it goes helicase, then it goes topoisomerase. As you can see right here. Yeah, DHT. Yeah. Yo, don't we need to know like DNA pol 1 and DNA pol 3 and the differences? I don't think so. I'm not too sure about what pol 1. I haven't seen that on the CED. I don't think so, but well, let me know. Ask me again later. But I don't think we do. Primary signal to start DNA duplication. Yes. Yeah, okay. I'll try to wrap this up. What about the single strand binding proteins? At what point do they stabilize the DNA? Are you talking about topoisomerase? Alright. You guys, are you talking about topoisomerase and how it stabilizes the DNA? Right here? These right here? Single strand binding protein. Oh, okay. You don't need to know this. You don't need to know this. Yeah. Okay, alright, guys, don't worry. I promised that I wouldn't spend more than 90 minutes, so I'm gonna move on. Alright, practice MCQ. Let's spend 60 seconds on it. Yeah. Alright, so practice MCQ. During DNA replication, which enzyme relieves the tension in the DNA helix ahead of the replication fork caused by unwinding? Alright guys, I took this from Quizlet, so I took this from, in air quotes, Quizlet. So it should be an actual AP question. So, alright, let's see. Alright, let me unwind. Alright, so C. Alright, so, oh shoot, okay, wait. Do you guys want me to go over this, or? Okay, alright. I won't go over it, but in a nutshell, helicase unwinds it, DNA polymerase synthesizes new DNA, ligase glues the Okazaki fragments, topoisomerase unwinds it. Alright. Okay, if it's too, okay, alright, yeah, let's move on then. Okay, so, I expected a lot of people to leave, but not a lot of people have left, so please fill out this form. I'm only going to say this once, so please fill out that form. If you have to leave, fill out that form. If not, then let's continue. That form just gives me feedback as a lecture. But if you have to leave, fill that out. But if you want to stick around, let's go. All right, here's the course exam description for 6.3. All right. So again, if you don't, I'll go over this in depth, and let's. Okay, we don't need to know the different DNA polymerases, right? No. All right. Sorry, Gamer3. I just want to answer your question. I know I promised to get it wrong. Is this right? Polymerase 3. You don't need to know the different polymerases in the CAD, but you're right about ligase. It seals everything up. All right. So if there's no questions, I'm gonna head on to the next slide. So here's the overview of the genetic information flow. It goes DNA, then goes RNA, and that's proteins. Now that's just the basic overview. I'm gonna go through each step thoroughly. So this is called the central dogma of molecular biology, this DNA to RNA to protein thing. And transcription is the very first step. This is the process where we take the information from DNA and use it to make messenger RNA or mRNA molecule. Again, I'll go all over this in depth. All right, I'm gonna play a 60-second clip which has amazing animations, once again, to help catalyze the understanding.
To do this, RNA polymerase, with the help of protein...
Okay, just to get some context, they're talking about transcription. This is like, he's going to cover 6.2 first. So, I'll start 5 seconds back. Why is my slideshow doing that?
To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific
We don't need to know transcription factors yet. That's next.
sequence within the gene, which is called the promoter, and pries the two strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA.
Again, I will go over all these terms in depth. This is just an overview video.
And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from the gene and the DNA is returned to its original state. But we have produced an mRNA. This carries with it the information encoded in the gene, and after a few quick modifications during RNA processing, it will leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome. This is where...
Alright, so that was just a brief overview of this entire subtopic. And that video just had great animations, and I will be covering everything that video covered in depth. So, let's walk through this process step by step. So,
to do this...
What? Alright, okay. So, first of all, there's 3 different RNA molecules. There's mRNA, which carries information from the DNA to the ribosomes. There's tRNA, which brings amino acids to the ribosome during translation. And there's rRNA, or ribosomal RNA, same thing, which makes up part of the structure of ribosomes and help catalyzes protein synthesis. And as you can see right here, this diagram illustrates it perfectly. mRNA encodes for proteins, tRNA has a funky structure, it carries amino acids, and rRNA, or ribosomal RNA, carries and forms up the ribosomes. Alright, let me check Discord. Alright, oh, no questions. Alright, bet. Alright, so, I'm gonna need the laser pointer for this one, but let's do this. So, here's the step-by-step. So, during transcription, RNA polymerases only uses one strand of DNA. This is called the template strand. And here's the template strand right here. This is the template strand. And here's what I mean by template strand. The template strand, also called the antisense strand, is the strand that an RNA molecule is made based off of. As you can see, this is the template strand. This is the non-template strand, or non-template. And this is the template strand. As you can see, the RNA molecule is made using off of it, using base-to-base pairing. And RNA polymerase reads the template strand. Let's say RNA polymerase is like right here, right? It reads this template strand and builds the RNA strand in the 5' to 3' direction. The coding strand, this is the coding strand. The coding strand is the non-template strand, is the strand that is not being read. Again, the coding strand, despite its name, is not the strand being read. It will match the RNA sequence, except that all thymines are replaced with uracils. And here is why it matches the RNA sequence. As you can see right here, this is the coding strand, this is the coding strand, this is the RNA. It matches it exactly, except T is replaced with U, as you can see right here. And why is that? This is because of base-by-base pairing. T always pairs with A always pairs with the T or U in this case, C always pairs with the G. And that's why the RNA will always match the coding strand, except all thymines in the coding strand are replaced with the uracil. Alright, let me open Discord. I sent a lot of questions for that. Alright, let me catch up. Aren't there like 20 different terms for coding? Yes, there are. Oh my gosh. I don't even want to get into that, but I looked at the CED. The template strand is either called the antisense strand or the template strand. The coding strand is just called the coding strand or the sense strand. Alright. Alright. Am I the only one who doesn't get this slide? Okay, so let me explain this briefly again. So, actually, sorry. Sorry. Okay. Let me. I'll get back to you, and I'll see if I don't have a lot of questions. I'll re-explain it again. But don't we also need to know all the places transcription takes place in? If you're talking about prokaryotes and eukaryotes, yes. But yeah. Oh no, it's all good. Yeah. Sorry. Wait. Sorry. What's the difference between coding and template? Oh, so this is the template strand. This is the template strand of DNA right here. This is the template strand. And this is the coding strand. Wait, you guys can see my red marks, right? Okay, you can. Perfect. This is the template strand. Sorry, this right here is the template strand. This right here is the coding strand. Coding strand. And the difference is that the template strand is red while the coding strand is not red. Yeah, so the diagram is overwhelming. So yeah, I completely understand. Yeah, this is why I spent a long time on this. I will be publishing the slideshow, and the video I published was really good. So, but yeah, I'll try to answer my questions as best as you can. All right, sorry, let me catch up on Discord. Wait, do APZNs differ per state? No. Do they differ per state? Well, there's different versions, so not per state, but they do have different versions. I thought we were doing DNA polymerase. Why is there RNA polymerase now? So DNA polymerase, that was for DNA replication. RNA polymerase, that is for the formation of mRNA using transcription. Wait, is the template strand only from 3' or 5'? So, okay, so it gets read. It gets read 3' to 5', but it gets built 5' to 3'. So DNA can only synthesize from 5' to 3', but it gets read from 3' to 5'. Where the hell did the U come from? So U, that's uracil, and uracil replaces thymine. Uracil replaces thymine in RNA. RNA has uracil U, while DNA has thymine T. And that's the main difference between, that's one of the main differences between RNA and DNA. Oh, sorry. Is the RNA polymerase the enzyme that splits the two RNA strands? Yes, in the video, yes. Sorry, well, the RNA polymerase is the enzyme that splits the DNA strands. It splits the DNA strand and forms mRNA. It's a template strand. Yeah, all good, all good. You want to form. Okay, so here's why you want to form mRNA. I'll get to that later, but the reason why we need mRNA is so we can synthesize proteins, and I'll cover that in the translation side. So the coding strands are the ones being read by RNA polymerase. Sorry, the coding strand. This is where it gets confusing, Moose. The coding strand, confusingly, is the one not read. The one being read is the template strand. The coding strand is not involved in the RNA replication at all. This is the template strand is the one being read. I thought we were making DNA right now. Yeah, we're making. Well, sorry, no, we're not. We're making RNA right now. So the coding strand is the mRNA strand. Yes, the coding strand, if you want to think about it that way, is the mRNA strand, and the template. No, sorry. So the coding strand is not the mRNA strand, but it matches it up base for base. This is the RNA strand and the coding strand are different. This is the RNA strand. This is the coding strand. The coding strand just matches this RNA molecule, base for base, except T is replaced with U. And the template is kind of intermediate. I mean, I guess you can think about it that way, but RNA polymerase just reads the template strand. Yeah, you're right. This is transcription. Can you mark the 3' and 5'? Yes. So it gets read 3' to 5', right? So let's say this is RNA polymerase right here. DNA is always synthesized. So this right here, this is 5. Oh wait, this A right here is at 5', and this U right here is at 3'. But this T right here is at 3', and this second T right here is at 5'. And that's why I said that it reads from 3'. That's why I said that RNA polymerase reads it from 3' to 5', because as you can see, let's say this is RNA polymerase right here, this weird looking thing. It reads it 3' to 5', but it builds it 5' to 3'. Yeah. Do you guys have helpful things to remember this stuff? I will. Yeah. I mainly use Anki, and I'll publish that. I'll publish my personal deck. I think Anki is the best tool to ever grace this earth for memorization, and I'll publish my deck at the end of this lecture and also online. But yeah, I'll do it later. So transcription makes an RNA strand, and a translation makes yes, alien. That's completely right. How does RNA polymerase split DNA? Helicase and RNA polymerase both split DNA, but they both do it for two. But RNA polymerase splits DNA to form mRNA, while helicase splits DNA to form DNA. Can you explain what occurs when the Tata box is met? When the Tata box? Sorry, wait, hang on, let me catch up. Wait, Adams, what do you mean by the Tata box? Alright, sorry, I'll move on. But yeah, I'm just reading a Discord from top down. Alright. The binding site of transcription factors. Oh, yeah. Okay, so transcription factors, that'll be in part 2. And I do actually know what you mean by Tata box now. Yeah. But that'll be in part 2. So, RNA polymerase splits DNA. Okay, here's why I'm hesitant to say yes to that. It splits the DNA strand. It splits the DNA strand. It breaks this right here. Look at this line right here. RNA polymerase splits DNA right here. But helicase also splits DNA. Helicase also splits DNA. But they're just for different reasons. You do not need to know the Tata box. I looked at the CD. You don't. RNA polymerase is used in transcription. RNA polymerase is just used in transcription. Helicase is only for DNA replication. Helicase is only for DNA replication. Yes, that's right. What splits RNA strands then? RNA is usually single-stranded, but AP biology doesn't really cover. Well, nothing really splits the RNA strands. Yeah. Okay. In part 1, there's only one more topic left. This will end at 6.4. All right. Yeah, I totally understand. Yeah, I'm sorry. I get caught up in the question. Sorry about that. Yeah. All right. I'm gonna move on. All right. So, here's the transcription process steps. So, here are the basic steps of transcription. First, RNA polymerase binds to the promoter region of DNA. This is the promoter region of DNA. Then it opens up the DNA. And then an RNA strand is synthesized on this template strand of DNA right here. RNA is right here. RNA polymerase is right here. It's going that way. It's synthesizing an RNA molecule based off this DNA molecule that it just unwound. And finally, once the RNA molecule is finished, it gets released and eventually synthesized into a protein, but I'll cover that later. Alright, let me check Discord. Alright, no questions. I'm gonna move on. Okay, I would say 6 is the hardest. Yeah, this is the hardest. Alright. Okay, and this looks really daunting, and this looks like a lot, but this is just pure memorization. There's no conception about it. Just get some flashcards and memorize this. But here are the mRNA transcript modifications. So in eukaryotes, before the initial mRNA transcript has to be modified before it can leave the nucleus. Basically, the mRNA has to be modified before it can leave the eukaryote nucleus. There are 3 major modifications which you need to know and you need to memorize. You don't need to know why they're there. You just need to know that they're there. So first, a GTP cap is added, and this helps with ribosome recognition and translation initiation. Next up, a 3' poly A tail is added, and this is a long adenine tail. Oh yeah, sorry, the GTP cap, that's guanine. The poly tail is a super long adenine nucleotide to add onto the 3' end of RNA. And this helps protect mRNA from degradation. And next up is the introns. And last lecture, people were very confused on this, so I made sure I'm gonna make sure to explain this really well. The third major change that you need to know is that the mRNA undergoes splicing. And splicing is when introns and exons are removed. And now, what are introns? Introns are the non-protein coding region of genes. So, DNA is made up of long nitrogenous bases, AT, GC, whatever, right? Not all of these DNA segments code for a protein. And that's what introns are. Introns are non-protein coding regions, while exons are protein coding regions. Let me know if that makes sense. Let me open Discord. Okay, wait, I see. Wait, so RNA polymerase doesn't have limitations? I'm not too... by limitations, I think... let me know what you mean by that. Isn't some of this earlier units, or am I tripping? Introns are from earlier units, but they play a role here. Yeah. Alternative splicing, let's go. Yes, alternative splicing is coming up. Exons can also be removed. Why do we have introns then? Yeah, that's exactly the question I've been looking for. So, introns exist because they play a role in regulating gene expression. So, this intron right here, why do we have introns if we remove them, right? Well, introns play a role in regulating gene expression. So what is gene and basically, what is gene expression? Basically, it regulates the transcription of other genes. So introns, they don't code for proteins, but they help code for some other tools that help gene expression. Alright, let's see. Yeah, alternative splicing. What I mean is it a continuous comparative DNA polymerase? Oh, I see what you mean. Yes, there is no leading or lagging strand for the RNA polymerase. Does every RNA modification have a GTP cap and a poly? Yes. Super dancer, yeah, that's right. Yeah. Alright, yeah. Alright. Yeah, alright. I'm gonna move on if there's no more questions. I'll actually wait for like 10 more seconds. Yeah, sleep. Yeah. Alright, I'm gonna move on. Alright. Alternate splicing. I heard some people in the chat were hyped about this. So, sometimes the same mRNA can be spliced in different ways. So, what is splicing? Right? This is what I mean by introns being removed and exons spliced. Introns are in the way. Introns need to be removed. But the removal of certain introns and retentions of certain exons can generate different versions of the resulting mRNA molecule. This is called alternative splicing. So, this can increase the diversity of proteins that an organism can produce. So, here's what alternative splicing is. So, let's say this is the pre-mRNA, and by pre-mRNA, this is just a term for the mRNA molecule before it gets spliced. So as you can see, there's two different paths this can take. It can either take out the green one, or it can either take out the yellow one, right? And essentially, two different mRNA molecules can be made from a single pre-mRNA molecule. And this diagram illustrates it perfectly. Like this one is missing the green one, this one is missing the yellow one. And then during translation, which is basically when these mRNA molecules are read, different polypeptides and proteins can be made from a single pre-mRNA. So basically, that's the key of alternative splicing, and that's the main takeaway you should know. That a single pre-mRNA molecule before the removal of introns and exon splicing can produce multiple different proteins. And this helps increase the diversity of proteins that organism can produce. Alright, let's check this one. So, can you elaborate on what that sentence means? Yeah, retention is keeping. Sorry, Esen, can you say that? Sorry, I'm only on one monitor, so I've only been able to read Discord after. But not that deep, bro. Alright. Oh, sorry, Esen, I just saw you said, can you elaborate what that sentence means? Oh, Crest, so is this how mRNA gets made? Oh yeah, all good. So is this how mRNA gets made? I'm sorry, I zoned out. So, no. So this is this slide right here is how mRNA gets made using RNA polymerase. This slide right here is just a different concept known as alternative splicing, which essentially states that a single pre-mRNA molecule can form multiple different proteins. And by pre-mRNA, that's before the splicing is done. So back to Cress's question. I don't want to move on. So the pre-mRNA can form multiple different proteins. So alternative splicing is used after translation. Alternative splicing is used after transcription, but before translation. Translation is the reading of an mRNA molecule to form proteins. Alright. Does that make sense? Alright, cool. Alright, sorry guys. We're almost done, guys. One more subtopic left. This is the summary. Again, RNA polymerase, we need to know what that is. Template strand, this is the template strand. mRNA, tRNA, and RNA, we need to know those three different types of RNA, right? mRNA is used to transfer information from the DNA to the ribosomes. tRNA, I'm going to cover tRNA in a sec, but it carries amino acids. And I'm going to cover tRNA translation. rRNA, it just makes up the ribosomes. And for the main three things that you just need to memorize, the three main ways it's just processed, a GTP cap is added, a poly A tail is added, and it's spliced. So this is a review. Any questions? Yeah. I forgot. I know. Yeah, 8:30. I'm sorry, guys. We're almost done. Already forgot look out. Yeah. Fails 8:30. It's part two. Part 2 is gonna be 7 days from now. At this exact same time. Why add it if it will be sliced? Again. Yeah. So it's added because introns are not protein coding regions, but they're still used for gene regulation. They're used for various parts. Part 2, I'll publish part 2. I'll ping everyone part 2 in a few days. But yeah, it will be 7 days from now. Yeah. All right. Any more questions? Isn't there 8 units? Yes, there's 8 units. When you say RNA makes up the... Okay. It's just that rRNA makes up the ribosome. So rRNA is completely... rRNA is just an RNA molecule that makes up the ribosomes. You don't need to know anything more than that. RNA just makes up the ribosomes along with some other proteins. And that's just something that you don't need to know too much about, but you just need to know that RNA, yeah. Are introns used to make tRNA? That's a good question, but in the CED, the only thing they added is that introns were used for gene regulation. So I'm not too sure about that, but I I don't think that's something that you'll be tested on. Where will the slideshow be sent? It'll be sent online on the lecture guide. I'll be publishing this on the lecture. I'll probably be publishing this on the main Google Sheet that has all the lectures. Lance pinged at everyone a few weeks ago. Lecture guide, it's not up yet. All right. Do the entrons end up just like floating around? That's a good question, actually. You know, that's actually, I actually haven't thought of that, but I don't know the answer to that, and but I don't think that you need to know what happens if the entrons happen. Yeah. Where can you find the spreadsheet? Actually, okay. Sorry, I hate like keeping you guys here, but okay. Really quickly, here's the lecture guide. Yeah. All right. So again, thank you, Super Dancer, for posting as well. All right. So here is the practice MCQ. Let me read it out loud. Don't worry, this is a lot easier than it looks. Which of the following best explains how the structure of a eukaryotic gene allows it to code for multiple polypeptides? A. Each codon in the mRNA transcript can pair with multiple amino acids during translation. B. RNA polymerase can transcribe both the coding and non-coding strands of DNA simultaneously. C. The presence of multiple RNA polymerases on a single gene allows it to produce multiple mRNA copies. And D. Alternate splicing of the pre-mRNA allows different combinations of exons to be included in the final transcript. Alright. Okay, and this was an actual AP question that I saw, and you actually yeah, I'll let you answer. I'll explain after. All right. Oh, everyone's typing. Literally. All right. Looks like might. Oh yeah, I like that, everyone. Yeah, the answer is D. And this is I want to say that this one's a little tricky, so I'll go over why the right answers are right and why the wrong answers are wrong. So, the answer is D. And before I go over the right answer, let me explain why the wrong answers are wrong. So, here's why A is wrong. Each codon in the mRNA transcript can pair with multiple amino acids during translation. This is incorrect. First of all, this codons, which I realized I'll cover in the next subtopic. This is just a wrong concept, and I just realized that this is next subtopic. So this is just wrong. RNA polymerase can transcribe both the non-coding and coding strands of DNA simultaneously. Here's why this is wrong. RNA polymerase only transcribe the template / non-coding strand into RNA. It does not transcribe both at the same time. And again, it's not like helicase. RNA polymerase only transcribes one strand at a time. And okay, even if it did, which it doesn't, this wouldn't explain making different polypeptides for one gene. Like even if B was right, this doesn't really answer the question anyways, right? Alright, so it's C. The presence of multiple RNA molecules on a single gene allows it to produce multiple copies. And so, while it's true that many RNA polymerases can transcribe a gene at once, but by that I mean let's say there's multiple different DNA molecules. There can be multiple RNA polymerases. This just makes more copies of the same mRNA, and it doesn't actually create different polypeptides, which is what the question's asking for. And so here's why D is right? D is right because alternate splicing of the pre-mRNA, this just allows the different combinations of exons to be included in the final transcript. So in eukaryotes, a single pre-mRNA, which is the unprocessed transcript, contains exons and introns, which are introns, of course, are in the way. They're non-protein coding regions. And during RNA processing, the introns are removed, and different combinations of introns and exons that are kept and removed can produce different mRNA molecules based on the alternate splicing. All right, let me open Discord. Let me see if I have questions. What the logic? Oh, yeah, everyone. DNA equals DNA starts with D. Yeah, that's funny. All right. Wait, how is A wrong? So, wait, sorry, did I answer your question? One codon can only be trained. Oh, yeah, I won't, I'll answer your question about A later. because I just realized that codons are in the next subtopic. So. Oh, I'm sorry, Jen. Don't worry. It's 8:37 p.m. for me. It's 8:37 for me. Don't worry. Don't worry. Guys, this is the last. Okay. Guys. Any questions? Any questions about this? Last one, guys. Last one, guys. This is the very last one. All right. All right. No more questions. All right. I'm gonna move on. Okay. Okay. I say this is the last one. We might be here for another 20 minutes. This translation is very similar to transcription in terms of the difficulty. Yeah, probably not. Don't worry, guys. But I'm covering it thoroughly. Let's do this. So, let's get started. So, I'm gonna revisit this video. I'm gonna play it for 60 seconds. Let's I'll turn up the pen. Yeah.
The cytoplasm.
Okay, this video starts off where we left off. The first part of the video covered transcription. This part of the video will cover translation, which is the process by which an mRNA turns into a protein. Let's get
the cytoplasm, where it will find.
Oh my gosh, what's
The cytoplasm, where it will find a.
Okay, so basically, just to give a recap, this gold thing is the mRNA molecule, and it leaves the nucleus, and it goes to the ribosome. And this is where the video leaves off.
Ribosome. This is where translation occurs. During translation, the mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid.
All right, so that's the core thing. I'm actually going to go back 10 seconds to rewatch that.
The arrangement of the nucleotides bases on the mRNA, which we call cytoplasm, where it will find a ribosome. The mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid. The arrangement of the nucleotides into these codons is called the reading frame. Since there are 4 bases and each codon has 3 letters, 4 cubed gives us 64 different possible codons, which is more than enough to code for all the amino acids we need. The cytoplasm.
All right, so that was just a basic overview of translation. Again, I'll cover that. That video was just to give it an introduction. Let's actually get into the bread and butter, as some would say. All right. Okay, let me open Discord. So anticodons are of the coding strand, and codons are. Okay, so anticodons are completely different from the coding strand. Anticodons are carried by tRNA, and the codons are. mRNA contains the codons. Yeah. Hopefully, I'll cover it, and hopefully, I'll answer your question later. All right.
The cytoplasm.
Why is the video playing? Okay. Okay. So here's the very basic overview of translation. Translation is the process that converts mRNA sequence into a polypeptide chain, which will fold into a functional protein later. It occurs on the ribosomes in the cytoplasm, or that rough endoplasmic reticulum, which rough ER, if you don't know what that is, the rough endoplasmic reticulum is from Unit 2, so I won't be covering that too much. And there are 3 main stages of translation. Initiation, which is the start of translation. Elongation, which is the process by which mRNA is read and the protein is synthesized. And termination, which is when translation ends and the protein is finished. Okay, so Alien, will they make us translate sequence on the exam? Yes, they will, but it will be very, very easy. They'll give you a table, given a list of codons, and they'll tell you that ACG, ACA codes for, I don't know, what's an amino acid name? ARG or something. And so they will make you translate a sequence on the exam. And, but don't worry, those questions shouldn't be too hard if you just practice enough. Alright, Crest, you're right, yeah. So here's a little bit more about ribosomes. So prokaryotes. In prokaryotic cells, here's the key. Translation begins while the mRNA is still being transcribed, and I'll talk more about what that means in a sec. And in eukaryotic cells, transcription happens in the nucleus, and translation happens later in the cytoplasm or on the rough ER. And so here's what I mean by that. So let me get my pen out. This is the prokaryotic cell. And remember, prokaryotes do not have a nucleus. Eukaryotes have a nucleus. Prokaryotes do not have a nucleus. Prokaryotes do not have a nucleus. And so because prokaryotes do not have a nucleus, transcription, which is the process by which a DNA molecule is used to form an mRNA molecule, occurs during the same time as translation. Let's take a look right here. Let me get my pen out. This is the prokaryotic DNA. Transcription is occurring here. This purple thing, this is the mRNA molecule. Transcription is happening right there. But at the same time while transcription is occurring, the ribosomes, these little jelly beans right here, they're also doing translation at the same time as transcription. So I'll say this. Transcription happens during the same time as translation only in prokaryotes. Only in prokaryotes. And I'm emphasizing this a lot because I've seen this a lot on MCQs. Eukaryotes, they happen separately. As you can see here, this is the nucleus. Transcription turns the DNA. No, sorry, it doesn't turn it. Transcription forms an mRNA, a pre-mRNA, from a DNA molecule. And this is not transcription. Then it leaves the nucleus, and then the mRNA is used by the ribosomes to make a protein. As you can see, here's the distinct difference. In this one, they occur at the same time. Transcription and translation occur at the same time. In this one, they occur at different times. Alright, and just as a reminder, ribosomes consist of rRNA and some other proteins. Alright, let me open Discord. So prokaryotes are more efficient. So the process is not necessarily in terms of efficiency. Prokaryotes, it just happens naturally because they don't have a nucleus. No. Yeah, for sure. Okay, sorry. Another question? Okay, sorry. I feel like that was really complicated, so I'll actually stop for questions. Anyone? Alright. Oh, cool. Alright. Again, I'm emphasizing this because this is something that you'll probably see a lot on the exam. The fact that the main difference between prokaryotes is that transcription and translation occurs at the same times. And eukaryotes, they happen separately. Alright, let's move on. Actually, wait, I see you posted a diagram. Yeah, that's good, yeah. Alright, so, now here is where anticodons and tRNA come into place. So, I have an animation that I'll play after this, but here's the overview. Each tRNA molecule has an anticodon that is complementary to the mRNA codon. And a reminder, codons are a set of three nitrogenous bases. A, this right here, that's a codon. U, that's an anticodon. This is a codon. This is anticodon. And the main difference between the two is that the codon, the anticodon, is on this molecule right here called the tRNA. And the main relationship is that the tRNA's anticodon will match up with the codon of the mRNA. As you can see, they're complementary. We all know that adenine binds with uracil like this, right? We can see that guanine pairs with cytosine just like that. As you can see, they're complementary, and that's the key. And tRNA, upon recognizing that the anticodon, upon the anticodon recognizing that this is its pair, right? It will connect this amino acid right there, growing the polypeptide chain. So that's the key. All right, I won't answer any questions about this because I have a great video that will show this.
Let's see the process in action.
This is just the process of translation as a whole, and these animations are great. This animation, I didn't want to focus on it because it contains a lot of unneeded terms, but all the terms I've stated so far is all you need to know. You don't need to know any other term than what I stated so far that this video will say. But just focus on the animations.
Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Alright, so that video right there showcased exactly what this had right here. Let's go back. This showcased how tRNA anticodon recognizes a codon and adds an amino acid onto the growing polypeptide chain. Okay, so how much longer is this? Guys, we're almost there. This is the very last topic. I think there's 4 slides left. Alright, and this goes with initiation. Ribosome binds with mRNA at the start codon, AUG. And you don't need to memorize AUG. You just need to know what the start codon is. And that ribosome binds to mRNA at that start codon. Elongation. tRNA brings more amino acids, and peptide bonds form as the amino acids bind to the polypeptide chain. And determination. The stop codon, it signals the start of the polypeptide. Alright. You're done, guys. We're almost there. I'm so sorry. But, good thing I separated this into parts. Alright, next slide. Alright, so, actually, before I get into this concept of this slide, I want to revisit that question I said I would revisit later. To that person who asked, would they ask me to transcribe a sequence of codons to amino acids? And yes, they would. Yes, they would. I won't cover that, exactly how to solve those problems, but as long as you do a couple practice problems, you'll see it's super simple. All you have to do is just read a table like this. It will either look like this, or it will be a table. And it's I promise you guys, it's easy. Sorry, I don't want to say it's easy, but it's very intuitive. And if you do a couple practice problems, you'll get the hang of it. So, back to actually what this slide is saying. The genetic code is almost universal. And what do I mean by that? Almost all living organisms on this entire planet use the same codon to amino acid assignments. what does that mean? So let's look at this example right here. GAU. Let's look at GAU. That will always GAU codon will always correspond to the amino acid ASP. Always. It will always will be. And the fact that all organisms have the exact same codon to amino acid assignment is such good evidence that all life shares a single common ancestor. We probably all came from a single bacteria. That's just something we need to know. Alright, next slide. Okay, so actually I'm gonna say any questions because I'm gonna move on to a completely different topic. Yeah.
It's awesome.
Next up is retroviruses. For those who, for my fellow retroviruses out there, I'll wait for some questions. All right. Yeah, retroviruses. All right. I'm actually okay. Yeah, you can just keep. I see some people are typing. You can type your question in. I'll answer it later. But I'm gonna be moving on to retroviruses. So, retroviruses. They're very they're a lot to memorize, but they're also very intuitive. Let's pull out the whiteboard for this. Retroviruses are special viruses, and they're like HIV. If you know what HIV is, they're a retrovirus. And they reverse the typical information flow. So here, all you need to know for the AP exam is how retroviruses work. And even then, the last time I saw a retrovirus MCQ or FRQ was ages ago. But you still need to know this. It's on the CD. So the retroviruses reverse the typical flow. They turn RNA to DNA. Normally, cells turn DNA to RNA. Retroviruses turn RNA into DNA. So here's the steps, right? So first of all, retroviruses use reverse transcriptase, which is an enzyme that catalyzes reverse transcription. Basically, reverse transcriptase and reverse transcription turn RNA to DNA. That's all you need to know. Reverse transcription is the process by which RNA is turned into DNA. And now here's where retroviruses get tricky. Retroviruses insert their DNA, this DNA, into the host cell's genome. Let's take a look at this right here. This is the RNA, right? It's an RNA molecule. That's all it is. It's just an RNA molecule. Then through reverse transcription, this RNA molecule is turned into DNA. Then it inserts this DNA into the target cell's DNA, into your DNA or something. And afterwards, this DNA gets transcribed, and then this viral RNA, this is the imposter, right? This is the imposter. It goes to the ribosomes, and it's used to make. Oh, this step isn't shown in this diagram, but this viral RNA is used to make imposter proteins, and these proteins are then used to make new viruses, which then repeat the step all over again. And so basically, these new viruses leave and infect more cells. So, just to recap exactly what I just said. The viral RNA turns RNA to DNA. Then, this DNA gets used to make viral RNA, imposter RNA. And this is used to make proteins. And these imposter proteins are used to make new viruses, and these new viruses infect more of cells. Any questions about that? Let me open Discord. Spoiler, unaltered memorization. Oh yeah, call me Hellcase the way I want, unzip your genes. Yeah. I remember my teacher said that. Will the AP exam ever try to confuse us in terms of this translation problems? Like switching up how the codons are presented? Yes. Oh my gosh. Guys, everyone, that's a very good question. They will do that. And I actually remember I got a question wrong because they swapped out the 5' to 3'. Yes, they will do that. They will do that. Why do viruses want to kill us? Well, they don't want to kill us. They just want to survive. And sadly, they just do that by killing us. Yeah. Imposter, oh my gosh. I knew the word I picked, imposter, was cooked. My teacher never taught us this deeply. Okay, and I think that's fair on your teacher's part. I've only seen like maybe 3 MCQs in all of my AP biology years that actually like tested this. Will we have to know lytic versus lysogenic cycles? No. Your teacher said that to you? Okay, so my, if you're talking about me, my teacher, well, no, just in my experiences, I've taken like probably 3 full-length practice exams and like almost in order to review for this, but yeah. So is it like HIV when it affects the cell and produces viruses from inside the cell? Yes. Lytic versus lysogenic cycles, going back to that, I've never heard of that, but I looked at the CED and I don't think so. Is this the end? Oh, guys, we're almost there. Summary, guys. This is the... Guys, stick around, please, for 10 more minutes, because please fill out the form. But anyways, here's the summary. Translation makes proteins, polypeptides. Proteins are polypeptides for mRNA using ribosomes. The codons are 3 nucleotide sequences that specify amino acids. tRNA molecules bring the correct amino acids to the ribosomes. And then translation proceeds, goes in 3 parts. Translation occurs in distinct stages: initiation, elongation, termination. Those are 3 steps. You know that. Another thing that wasn't said in this slide, but so you know, is the universality of the genetic code points towards a common ancestor, and retroviruses reverse the normal flow of genetic information. Alright, so before the very last practice MCQ, any questions? Guys, we're almost done. Okay. Thanks, guys. Alright. Okay, any questions? The last MCQ, guys, you got this. And, yeah, Unit 6 is really hard. Alright, last MCQ. And, I'm actually kind of regretting this, because this MCQ was actually... I put this MCQ because I got it, because I thought it was hard, so... Hopefully your brain's still working enough for this last one. But, alright, here's the very last MCQ. Oh, actually, I see a bunch of people are typing. I'm gonna assume that those aren't questions. I'll answer them later. Alright, here's the very last MCQ. And okay, let me read it first. Which of the following best explains a key difference in prokaryotes and eukaryotes with regard to transcription and translation? A. The 2 processes will occur simultaneously in prokaryotes but not eukaryotes. B. Prokaryotic mRNA is shorter than eukaryotic mRNA. Eukaryotic mRNA contains more coding regions than prokaryotic DNA. The processing of mRNA by eukaryotes is required for the mRNA to leave the nucleus. Alright, and, sorry, before I open Discord, the reason I thought this was hard is because technically two of these answer choices are true, but only one actually answers the question. Alright, let me open Discord. UC2, your voice is the first thing I've heard today. I'm honored. Wait, but isn't it like midnight? So it's the only voice you've heard today? Alright. Oh, okay, wait, okay. Alright, never mind. I thought this question was going to be really hard. Okay, yeah. The answer's... yeah, that... Yes, okay. So, yeah, but really shout out to you guys who caught that D is also right, but A is the right answer to this AP problem. So, let's go over why the right answers are right and why the wrong answers are wrong. Again, A is the right answer. But let's go over the correct answer first. So again, I emphasize this a lot. Prokaryote transcription will occur simultaneously in prokaryotes, but not eukaryotes. That's because, again, prokaryotes do not have a nucleus. While eukaryotes have a nucleus. Alright, and here's why B is wrong. Okay, and this just doesn't answer the question. B is, according to the AP College Board explanation, B is technically true, but it just doesn't answer the question, right? C, eukaryotic mRNA contains more coding regions than prokaryotic DNA. C is very misleading. Prokaryotic mRNA is, prokaryotic mRNA can actually code multiple proteins for one mRNA transcript, but that's just way beyond the scope, and C just doesn't answer it. And here's why D is also... The main distractor here is D. And it's true. mRNA is required for the mRNA to leave the nucleus. The GTP cap is needed, the poly A tail is needed. But that just doesn't answer the question of why the main difference in prokaryotes and eukaryotes are. So that's why A is the right answer. Alright, any questions about that? Yeah, D doesn't actually answer the question, really. The poly A tail is the long adenine, it's one of the modifications that is added onto the mRNA right after transcription, but before translation. It helps the mRNA stay, it helps it stay not degrade. Okay, wait, guys, don't leave. Guys. Okay, really quick, this is what you learned today. But before anyone leaves, guys, you're gonna need to fill out this form, guys. This form actually helps me so much. And okay, first of all, Master Lance wants us to fill it out. But also it does help me. Yeah. Okay, now, once you fill out that form, you can leave. But here is the full summary of everything you learned in this 2-hour span. You learned about the DNA and RNA structure. You learned about complementary base pairing. You learned about alright. You learned about complementary base pairing, and you learned about purines, you learned about pyrimidines, and you learned about how DNA and RNA structure differ. Second, we covered DNA replication. We emphasized that DNA is synthesized in the 5' to 3' direction, and that replication is semi-conservative. There's enzymes helicase, topoisomerase, DNA polymerase, and ligase. You need to know this. Third, we explored transcription and RNA processing. RNA polymerase binds to the DNA to create an RNA strand, and then the RNA is modified with 3 main things. You usually memorize these: 5' GTP cap, 3' poly A tail, the splicing of introns, which are in the way, and exons, which are protein coding regions. Those 3 things you need to know. And then fourth, we cover translation. mRNA is read by the ribosome to synthesize polypeptides. That is translation. We talked about initiation, elongation, and termination. Those are the 3 steps you need to know. And tRNA molecules bring the correct amino acids to the ribosome based on the codons. And so that is everything you've learned today. So, finally, I'm ending the recording. Any questions? I'm ending the recording right now, so you guys can talk if you want.